So far, we've talked about how enzymes are a class of proteins that fold up into a structure that has a particular active site. This active site is going to be what binds a substrate, which is our reactants in a chemical reaction. So our active site is going to bind this substrate, and then a chemical reaction will occur where the enzyme will help carry out the reaction. And in this depiction here, the substrate has been hydrolyzed into two different molecules. So we say that enzymes are biological catalysts because they speed up this reaction. A reaction that would normally take a very long time to happen, the enzyme is going to make it possible. By binding it in its active site, it'll help speed up the reaction so that it can exist in timescales that are relevant for life. All right, so what do we mean that enzymes carry out these reactions? Well, let's consider a reaction that we've been talking about, which is the hydrolysis of a triacylglycerol. Up to this point, we have talked about this reaction happening in the presence of acid or base, where the acid and the base participate in breaking these ester bonds, again, adding water across those ester bonds in order to form the components, which are glycerol and 3 fatty acid chains. So now what we're going to start seeing, now that we're moving into living organisms, these same reactions are now going to be carried out by our enzymes. And what you're going to see is that over these arrows, there is going to be an enzyme name. We know it's an enzyme because of the ending ase. And so this enzyme is going to bind our triacylglycerol, which in this reaction is the substrate. So lipase is going to bind this triacylglycerol. That means the triacylglycerol is our substrate. And then the reaction will happen in order to produce our products. Right, so now enzymes can be affected by temperature and by pH. They are very sensitive to changes in either one of these. So let's first consider temperature. So in this graph, we are looking at the reaction rate on the y-axis, and that is telling us how quickly a substrate is turned into product by the enzyme we are looking at. So that's just the rate of producing your product. In the x-axis, we have temperature. So what you're seeing here is that the first thing to notice is that there is a maximum here, meaning there is a temperature at which the reaction rate is the maximum. So right here at this temperature, all the way down here, so somewhere maybe around 37 degrees Celsius, that is going to be where our enzyme has the highest reaction rate. And again, you want your reactions to happen quickly in order for it to be relevant to sustain life. Okay, so we do have an optimum temperature, right? So what happens when we decrease that temperature? Well, when we decrease temperature in this direction, when we decrease the temperature going this way, what we see is that the reaction rate also decreases. And this happens because we know that in chemical reactions, if you decrease the temperature of a chemical reaction, you're going to slow down that reaction. We can also consider increasing the temperature. And what we see here is when we increase it in this direction, the reaction rate also decreases. But as you notice in this reaction curve, you see that the reaction rate decreases very dramatically. And this is because when we increase temperature, there's something else that is happening here. What is happening is that we disrupt the intermolecular forces that are keeping our enzyme together in the first place. So remember that the enzyme has to properly fold in order for the active site to be able to bind the substrate. If the enzyme does not properly fold, then it cannot bind the substrate, and therefore the reaction cannot happen. And how it folds is through these interactions of these intermolecular forces. We mean hydrogen bonding, salt bridges, and so on. So when we add heat to a reaction, when we increase the temperature, we start to disrupt those intermolecular forces. So what that means is that our protein begins to unfold. We call this denaturation. So we denature our protein by increasing the temperature. So what this means is that our protein is unfolding. It unfolds. And if it unfolds, well, the active site is no longer present, and therefore it cannot bind your substrate. And that is why your reaction rate decreases. Something similar happens with pH. So here I am showing you a graph of the reaction rate as related to pH. So what you see is that there is a maximum right here. And what this is telling you is that this enzyme, the optimum pH for this enzyme is maybe somewhere around 5. So that means that as long as your solution is around 5, your enzyme is going to have the highest reaction rate. If we decrease that pH, what you see is that the reaction rate decreases dramatically. And again, the reason for this is because when we change the pH, we are also disrupting or changing the intermolecular forces. we disrupt intermolecular forces. And again, whenever we do that, we also denature the protein. And so we are effectively doing the same thing when we increase the temperature or we change the pH. We essentially are disrupting intermolecular forces, and therefore our protein will denature, and the reaction of this enzyme can no longer proceed. Now, for enzymes and their optimal pH, that is going to depend on what tissues they are found in. So, for example, in our stomachs, we have a pH of around 2. So that means that the enzymes that are present in our stomach are going to have an optimal pH of around 2. So for example, in our stomach, we have this enzyme called pepsin, and the optimum pH for this enzyme is going to be somewhere around 2, because that's the pH at which it needs to function, which is in our stomach. Another example would be, for example, our intestines, which tend to be more basic. So our stomachs are more acidic. The intestines function at a more basic pH. So we have an enzyme in our intestines called alkaline phosphatase, and its optimum pH is going to be phosphatase, is going to be somewhere between 8 and 9. So its pH, optimal pH, is somewhere between 8 and 9. And again, this is because it must function in the basic environment of the intestines. So unlike the temperature, where you're going to see that most of the enzymes in our body, pretty much all of them, are going to function best at 37 degrees Celsius, which is our body temperature, we're going to see that the pH, optimum pH, is going to depend on which intestine it is in. And this is the reason why when you have a fever and your temperature is increasing, part of the danger here, the reason it's not good to have a fever, is because your proteins do begin to unfold, they begin to denature, and that can really affect the reactions that are happening in your body, which are important to sustain life. Okay, so these are ways in which the enzyme activity can be affected, where we're affecting the rate at which a reaction can happen, either by increasing temperature or changing pH. But there are two other ways, or actually other ways in which we can affect the activity of an enzyme, and this is called enzyme inhibition mechanisms. Now, this is going to differ from the changes in temperature and pH, because while the protein denatures or it unfolds when we change the temperature and the pH, in these mechanisms, the protein is not going to unfold. What is going to be different here is that we are going to add another substrate. We're going to add another molecule that is going to affect the reaction. So let's see how that's going to happen. In this course, we're only going to talk about reversible enzyme inhibition mechanisms. The first one is going to be competitive inhibition. So what we have talked about so far is shown here on the left-hand side. We have our enzyme with its active site, and then it'll bind a substrate. It's shown here in this green sphere. And then that is going to fit very nicely into our active site. And therefore, once it does that, the reaction can then proceed, and then you make products. So that's what we have talked about so far. Now, what is competitive inhibition? Competitive inhibition is when there is another molecule that is very similar in shape. So here we have this green spherical molecule, yellow spherical molecule that is really similar in shape. And this is necessary because how the competitive inhibitor works is that it's going to bind the active site as shown here. And so when the active site is occupied by this competitive inhibitor, now your substrate can no longer bind, and therefore the reaction cannot proceed. So in this direction, we cannot make products. So no products. And so what this means is that the substrate must compete for that active site. Now that molecule that's in the active site right now, as long as it's in there, this cannot enter the active site. When this competitive inhibitor comes off, since it is reversible, it's not going to stay there forever, it can come off. Once it comes out and the active site is available, then the substrate can bind and the reaction can proceed. But because it must compete for this active site, it will slow down the reaction that this enzyme normally carries out, because as long as the competitive inhibitor is sitting in there, there is no reaction that is happening. So that's what we mean by a competitive inhibitor. It's just some other molecule that's going to compete for access to that active site. Then we have another inhibition mechanism called non-competitive inhibition. So again, here I'm showing you on the left-hand side what we've talked about so far, with our green substrate being the reaction of interest. But now we're going to add a different molecule that does not have a similar shape. So because it does not have a similar shape, it cannot enter the active site. However, a non-competitive inhibitor can bind in a different part of the enzyme. And when it does that, the shape of your enzyme actually changes. in such a way to where the active site is no longer the shape that it needs to be in order for your substrate to bind the active site. So what this means is that even though this molecule is not binding straight into the active site, it's binding somewhere else on the molecule, that's going to cause a change in the entire conformation of your molecule that will affect the active site. And now your substrate just cannot enter there, and therefore the reaction cannot proceed. So we call this non-competitive because it's not competing for the active site, it's not entering there. It just changes the active site by binding somewhere else. So this will cause no products to happen, because in order for products to happen, your substrate must be able to enter the active site. And this is going to prevent the substrate from doing so. Now, why inhibition mechanisms are so important is because this is actually the tool that we use for drug design. So when we are trying to develop drugs, a lot of the drugs that we consume target some particular enzyme or a particular protein, but in this case, we're talking about enzymes. So you can design, if you want to stop a reaction from happening, for example, you can design a drug that can bind straight into your active site, and therefore you prevent that reaction from happening. You can also design a drug that binds somewhere else on the protein, but you get more or less the same effect, which is that you inhibit the reaction from proceeding. Now, of course, each approach has pros and cons, and depending on what you are targeting, it could have different side effects. But this is basically the main tool that we use to design drugs in order to deal with disease or manage symptoms.